minutes allowed for that. So at that point, please go ahead and turn on your video and microphone to ask a question or submit your questions using the chat feature at the bottom of your screen. And finally, we'll be recording and posting this talk on the CLP YouTube channel. So it's my pleasure to welcome Amy and take it away.
All right, thanks, Sheila. Can you all hear me? All right,
You're good.
Good. All right, so to the great disappointment of at least one of my lab members, this is not actually a chalk talk. This is a normal talk with slides. So sorry about that. So my lab is interested in bacteria that consume methane. And so that's what I'm going to talk to you about for the next 30 minutes or so. This is not working. Oh, there we go. So the kind of bacteria we work on in the lab are called methanotrophic bacteria, and these bacteria consume methane gas as their sole source of carbon and energy. And so on the center of this slide is the metabolism of these bacteria. So it all starts with methane. Methane is then converted to methanol, and then onward. And depending on the type of methanotrophic bacteria, the carbon is assimilated via different types of metabolisms. Methanotrophic bacteria are divided into 3 types: type 1, type 2, and type 3, and that's based on a number of features of their physiology, their morphology, the different metabolic pathways they use, and so on. So what I'm showing on this slide are just some micrographs of these bacteria along with the environments in which they're found. So the type 2 methanotrophs are found in like a lot of different environments. One common place they're found is in rice paddies where they consume methane. The type 1 methanotrophs are found in varied environments. The strain that we work on in our lab, M. capsulatus Bath, was actually first isolated in these hot springs in Bath, England. And then there are some unusual methanotrophs that live at high temperature and low pH, and they're found in crazy places like these volcanic mud vents in Italy. And so we're interested in how all these different types of methanotrophic bacteria are able to consume methane. So before I get into the details of what we do in the lab, let me just, sorry, there we go. Let me just tell you why you should be interested in bacteria that eat methane. So I think everyone here knows that methane is a potent greenhouse gas. It's second most important greenhouse gas next to carbon dioxide, and it's actually more potent than carbon dioxide over a 20-year time period. So I'm showing on this slide all different examples of methane in the news. So there have been a lot of methane leaks that have been in the news in the last few years. So for example, this little video here is showing a methane leak that happened in Ohio a few years ago. And basically the amount of methane released to the atmosphere from this leak was said to be sort of more than the man-made methane emissions from several European countries. So just huge. Down here is another methane leak. This one just happened this past fall in Pennsylvania. It was a company called Equitrans. And this methane leak actually erased, according to Bloomberg, 50% of the emissions gains from all the electric vehicle sales in the US. So there's all these issues going on with methane in the atmosphere. Another source of methane is agriculture and livestock. So this is a picture of a cow at the University of California, Davis, which has a big ag school. And what's going on here is they're actually measuring how much methane the cow is breathing out. So there's a big push in agriculture to make cows and other livestock like sheep exhale less methane, because livestock's actually like 14% of the methane emissions that we have. And so one way that this is being done is by feeding these cows different vitamins and supplements that actually suppress the amount of methane that they're exhaling. And down here on the slide is actually some beef that comes from cows. It's a company's called Lomi, or Low on Methane. And this is a Swedish company that is marketing its beef as coming from cows that are not emitting so much methane. So, you know, if you want to do your part, you could eat this kind of beef, although you could just not eat beef at all as well. So these are all the kinds of things going on in your daily world that have to do with methane. And, you know, I'm sure you've heard a lot about how we have to mitigate our emissions. One thing that's kind of special about methane is that we can actually take methane, convert it to methanol, and then use that to make useful chemicals and fuels and various feedstocks and things. And the thing is, like, methane's lifetime in the atmosphere is relatively short, about 10 to 12 years. So if we could channel all of this methane into some sort of process to turn it into something useful, that would be great. A big problem with that, though, is that the CH bond in methane is very difficult to break. And in industry, processes for direct oxidation of methane to methanol are high temperature, high pressure, very expensive. They typically involve conversion of methane first into syngas. So it's very difficult to come up with like a commercially viable way to convert methane to useful products. And that's where our friends in synthetic biology come in. So the idea is that, hey, let's take these bacteria that I just told you are out there in the environment and use them to take the methane gas, convert it to methanol in their first step of their metabolism, and then let's rig their various metabolic pathways to make all sorts of things that we might want to make. So that's great. But the problem is right here, this box in purple. In order to use a methanotroph to make methane into anything, you have to understand this first step, the conversion of methane to methanol. And it turns out that it's not that hard to, you know, take methanol and turn it into other things, but these bacteria don't do this conversion super quickly, and we don't understand on the molecular level how to make them do it better and faster. And so that's where our lab comes in. We're interested on the molecular level in understanding how these bacteria are able to break this strong CH bond in methane and convert it directly to methanol and not over-oxidize it. So the enzyme just converts it to methanol. And so this is the reaction of interest here. The enzymes in the methanotrophic bacteria that do this are called methane monooxygenases. So it takes methane and dioxygen, 2 electrons and 2 protons, and it makes methanol and water. So it's a monooxygenase because 1 atom of oxygen goes to make methanol, the other goes to water. And these bacteria have come up with 2 completely independent enzymatic solutions to accomplishing this reaction. One is a soluble enzyme, the soluble methane monooxygenase, and this is an enzyme that's found in the cytoplasm of these bacteria, and it uses a dinuclear iron center to perform this chemistry. This enzyme is only expressed by a subset of methanotrophic bacteria, and in those bacteria, it's only expressed when the bacteria are starved for copper. When the bacteria get a reasonable amount of copper in their environment or their growth media in the lab, they turn off expression of this one, or at least half or more of them don't even have the genes for this one, and they use this other enzyme called the particulate methane monooxygenase. And this is a membrane-bound enzyme. Particulate just means membrane-bound, and it consists of regions in the periplasm of the bacteria. So these methanotrophs are gram-negative, meaning they have an outer membrane and an inner membrane. And so this is at the periplasm, and then these helices are in actually what we call intracytoplasmic membranes. So this is what the bacteria look like when they're making this PMMO. And all of these stripes you see here are intracytoplasmic membranes that form by sort of invagination of the inner membrane, and they're all filled up with this PMMO enzyme. Now, unlike the SMMO, where we know exactly what the active site looks like, this diiron center with histidine and carboxylate ligands, for the PMMO, all we know is that it requires copper in its active site. And I won't go through the data we have proving that its activity is dependent on copper. I'll just ask you to believe that. And it kind of makes sense since its expression, its use in some bacteria depends on having copper in the environment. So basically all of our work for the past, couple decades has been directed toward this very basic question of what does the copper active site of this enzyme look like? Where is it in this trimeric structure? What is its environment? And then once we know that, maybe we could understand how to improve its activity and deploy it in some sorts of applications. Another thing I need to tell you is that this enzyme and everything I'm going to tell you about it is done on enzyme that we isolate from the native bacteria. So we grow these bacteria in a fermenter or a bioreactor with methane and oxygen. They make all these membranes, so the enzyme is essentially naturally overexpressed, and then we isolate the enzyme and study it. So we're getting it right from the native organism. It's not being expressed in E. coli or any other host. And so that means what we're looking at is very perhaps physiologically relevant, but we have not been able to do some basic biochemical things like make site-directed mutants and study them. And I think you'll see as I go through the story that that would be very useful. Okay, so let me tell you a little bit about the overall structure of PMMO. So I mentioned it's a trimer. Each trimer is made up of 3 copies of what we call the protomer. And the protomer has 1 copy each of the PMOB, PMOA, and PMOC subunits. PMOB consists of 2 paraplasmic cupridoxin-like domains and 2 transmembrane helices. PMOA is made up of transmembrane alpha helices, as is PMOC. In some, but not all PMMO structures, there's an additional alpha helix that we refer to as helix X that is not connected to the PMOC subunit and that we have not been able to identify by mass spec or structural approaches. It's not in all PMMOs, but it's in some, and we think it could be important given that we're isolating the PMMO right from the native bacteria. So as you see here, we've got some copper ions in this crystal structure. And so the way we've approached this problem is to look at the copper sites that we can see in the structures and think about whether any of those could be the site of methane and oxygen binding, the active site. So the first site I'm going to tell you about right here is what we call the copper B site. It's located in the PMOB subunit, just near the interface with the transmembrane regions. As shown here, it's got a single copper ion coordinated by 3 histidines, and this one histidine here, his29, is the amino terminal residue. So the first 28 residues are a signal sequence that gets cleaved off. And so both the amino terminus and the side chain of this histidine are ligands to this copper ion. Now, we know that this copper site is copper 2 in the cells, and we know that from a lot of EPR experiments that we've done in collaboration with Brian Hoffman, and I have some EPR spectra over here. So up top here is an EPR spectrum of whole cells, and what you can see here is that there's a single EPR signal attributable to copper 2. So EPR detects unpaired electrons. So copper 2 is a D9 for you inorganic chemists out there. So we have an unpaired electron. Copper 1, by contrast, is D10 and not detectable. The EPR signal has like these 4 bumps because of the nuclear spin of the copper. And we have a lot of corroborating EPR evidence that suggests that this signal here corresponds to this site here. We also have some data suggesting there's an axial water ligand there. Now, this site is conserved in the PMOB subunits from the types 1 and 2 methanotrophs, but not the type 3s, which are those unusual ones that grow at high temperature and low pH. Because it's not conserved in one of the types of methanotrophs, we think that it's unlikely to be the active site. Moreover, as we've done more and more spectroscopy on it, it's quite clear that it's always copper-2 and it's coordinatively saturated, which is kind of not consistent with having to bind and activate dioxygen for the monooxygenase reaction. Okay. So then the second site that I want to talk about is located here in the PMOC subunit, and we're calling that the copper C site. Now that site we know is copper 1 in vivo, and that's because the whole cells have just one EPR signal attributed to one copper 2, and we know that's from copper B. And then when we isolate membranes and then purify the enzyme, if you look carefully at these spectra, basically what you see are more bumps growing in. And what that means is that a second EPR signal is growing in. So what we believe happens is that as we take the enzyme out of the cell and start to purify it, this site, which starts out as copper 1, begins to oxidize to copper 2, and thus we get 2 different EPR signals. We also have evidence for an axial water near this site. Interestingly, this site can also be occupied by zinc. So if we crystallize the enzyme in the presence of zinc, which is sometimes needed to mediate contacts in the crystal lattice, we also see zinc in this site. This site, in contrast to copper B, is conserved in all methanotroph PMOC sequences. And based on several lines of evidence, our thinking, at least until the story that I'm going to tell you today, was that this is the active site of PMMO. And we had some evidence from native top-down mass spectrometry that we did with the Kelleher lab that increased copper loading of this PMOC subunit, although not necessarily this specific site, is correlated with activity. So we've known for several years that it's important to have copper in this subunit to have activity of PMMO. However, there's one big caveat with this site that was bothering us for a long time. And that's shown here. So it turns out that this site, copper C site, neighbors a region of the PMOC subunit that's about 30 residues and that is not visible in any PMMO crystal structure. And so that's shown here. So these residues between TRP226 and glycine 198, or I should have said it vice versa, are just disordered. There's no electron density. So it's kind of hard to make a case for some copper site being the active site when you can't really see its whole environment and what's going on around it. And that was compounded by this rather disturbing finding that we had after amassing more and more sequences of PMOC. What we found is that that region that we can't see in any crystal structure, which is shown here in this sequence logo, that region is the most highly conserved part of PMOC. So in these sequence logos, the big letters mean that things are highly conserved. And not only that, but these the sequence here contains several residues, including these 2 histidines and a couple of glutamic acids that look like they could potentially bind metal ions. So that's kind of a big caveat with our interpretation of the crystal structures and trying to discern the active site. A second big caveat with all of our work is the following. The crystal structures actually, I hate to say it, but it's true, don't really represent an enzymatically active form of the PMMO. So what happens is when you purify the PMMO in detergent, you lose a ton of activity. So what happens is we take the enzyme out of the native membranes, we surround it with detergent to solubilize it, to crystallize it, And if you look here, here are some numbers for methane oxidation activity, and you can see that the activity is very low on solubilized and purified enzyme. The reason that we have 2 bars in each of these experiments is that we have to reduce the enzyme to get activity. So presumably you need to reduce a copper 2 to copper 1 to react with dioxygen to make the species that can oxidize methane. And in vitro, we found that we can use duraquinol or NADH to do this. The actual physiological reducing pathway for PMMO is still under some debate. So in trying to get to the root of this loss of activity, one of our former graduate students, Sue Rowe, decided to try to put PMMO back in something more resembling the membrane. And so what Sue first did is put PMMO into bicels. And in a bicell, you surround the enzyme by lipids, and then the detergent goes around that. And what you can see here is that what she did, when she did that, we recover a significant amount of activity. So that told us that the membrane is quite important for the activity of the enzyme. Sue and Chris Koo in the lab then went on to put PMMO into another type of membrane mimetic environment, a nanodisc. And this is like the bicell in that you surround PMMO by lipids, but then instead of detergent, you have this membrane scaffold protein that surrounds it. And these are some data just showing activity of membranes and then of nanodiscs and of nanodiscs prepared in the presence of copper to show that we could get activity in these nanodiscs. And we originally did this actually to do the native top-down MS with the Kelleher lab, which was kind of annoying because you have to put it into a nanodisc just to blow it apart in the MS. But we quickly realized that this could be a useful way to study a more active form of the enzyme. And so then Frank Tucci in the lab tested the use of a bunch of different lipids in making these nanodiscs. And so here are some activity numbers that he obtained on a different preparation. So this is using detergent. And then these are nanodiscs with different lipids. So POPC and DMPC are what people typically use. Frank, in addition, made what we call native lipids. So he used like an old time biochemistry protocol to just isolate lipids from the native organism. And what he found was that that actually gave the best methane oxidation activity. So putting it back in a membrane mimetic environment with some lipids from the native organism helps with the activity. And so at this point, we thought, okay, maybe we can finally get a structure of the active PMMO, because we knew that you can actually use cryo-electron microscopy to characterize membrane proteins and nanodiscs. And so Chris Koo in the lab embarked on this heroic quest to get structures of PMMO in an active form in these native lipid nanodiscs. And I won't go into any of the details. I just want to point out, this is the Cryo-EM density map colored by resolution. And the overall resolution of the structure he got is 2.14 angstroms, which is actually much better than our best structure of PMMO and detergent at 2.6 angstroms. So our idea here was we're going to determine the structure and, see maybe what it is about the PMMO in these native lipid nanodiscs that makes it more active. And so this is the Cryo-EM map of the structure. Each protomer is shown in a different color, and this gray belt is the nanodisc. And so I'll strip that away. The overall structure is the same as what it was by crystallography. It's the same trimer. The overall folds of the subunits are the same. So I can strip away the map and show you the overall structure. It looks just like what I showed you at the beginning. And now what I'll do is I'll show you the metal centers. And we'll start with this paraplasmic copper B site, which we don't expect to be particularly different since it's in the paraplasm and soluble. So I'll zoom in. There we go. And this is the density. So this just gives you an idea of how well we can see things in this Cryo-EM density. We can rotate it around. We very clearly see the ligands and the copper there. We see density for 2 water molecules. These are actually closer to 3 angstroms away from the copper, so they're not coordinated in this structure. And I can contour the density a little differently. And this just gives you an idea what a well-occupied copper center looks like in a cryo-EM map. Okay. So that's the same as by crystallography. So now what we'll do is we'll move into the transmembrane region of the protein. So I'll zoom back out. And what we saw were many densities corresponding to lipids. So you can see those here in pink and blue. So you can see the head groups and the hydrophobic tails. So you see these sort of densities on top, and then these split densities for the tails of the lipids. So we were able to model about 36 lipids in the structure. And we could see very well-ordered lipids around the outside of the protein, and we could see them both on the periplasmic side, as shown here in pink, and then on the cytoplasmic side. And if we look on the inside of the PMMO trimer, we could see a very beautiful bilayer of lipids. And this was exciting to see because in the crystal structures, there's just kind of an open space in there. And we always thought it's probably filled by lipids, but here we can just see that quite beautifully. Okay, so now what I'm going to do is we're going to take a look inside of the trimer at this region here. And we're going to look at that where that copper C site I showed you was, and we're going to look and see if we can see that region that was disordered in all the crystal structures. So I'm going to zoom in, I'm turning it around. So now we're kind of inside the trimer, and we have this cluster of very well-ordered lipids. I'm going to strip away the PMO B and A subunits and just look at PMO C. And it turned out that all of this bright yellow part of PMOC is newly modeled in the cryo-EM structure. So this is the part of PMOC that we couldn't see in the crystal structures. And in the cryo-EM structure in the native lipid nanodiscs, it's stabilized by these lipids that are interacting with it. So that disordered region forms or completes 2 helices. So you can kind of see that there's a 4 helix bundle here. Before there was kind of nothing there, it just ended. And then we went to look for that copper C metal binding site. And to our shock, we saw a completely different metal binding site. And so what you're looking at here is this part here, this histidine, and this aspartic acid were the ligands to the copper C site. Instead of seeing metal there, we saw it here about 5 to 6 angstroms away, coordinated by 2 histidines and an asparagine residue. So this is where the copper C site was in the crystal structures, and this is a new site that we began calling copper D. Over here is just to remind you what the crystal structures looked like. So this is one of the structures where we had zinc and copper C, but I'm showing you it in the same orientation. So we had metal here and absolutely nothing here. Now we have all of this new polypeptide chain and a brand new metal center. So here's the density for that, and you can quite clearly see that there's density for a metal there in copper D and no density for metal in copper C, just density for a water molecule. I can strip that away, turn it around, and it was quite clear that there was a metal there. So here is just a still view comparing the crystal structure here with the cryo-EM structure. So all of this is new in the cryo-EM structure. I didn't even talk about this, but we were also able to see some more of the PMO-A subunit, including this arginine, which kind of sticks into this region where the copper binding sites are. And so this seemed crazy. And of course, I didn't believe it at first. And I made poor Chris get more cryo-EM structures, make more nanodiscs, collect more data. And he collected 4 data sets on the PMMO he was working on from this organism. And in 3 of the 4 data sets, this copper D site was occupied, and the copper C site was occupied only by a water molecule. And we know that's a water, not just based on the appearance of the density, but based on the distances to the surrounding residues, which are hydrogen bonding and not coordinating distances. In one of the structures, the water molecule had some density that might be copper C, and this part was disordered. So it seems like if everything is nicely ordered, we have metal here and not here. And if we get a little disorder, we might get metal there. Okay, so this raised a lot of questions, of course. One question was, is the copper C EPR signal actually attributable to copper D? So I showed you this before, and I said once we get the protein out of the cells, we get this second EPR signal, which we attributed to that copper C site. And indeed, we had data suggesting it was the copper C site. But the thing is, the ligands are almost the same. So for copper C, we have 2 histidines and an aspartic acid. And for copper D, we have 2 histidines and an asparagine. And using EPR and Endor with Brian's lab, we were able to see that there are 2 histidine ligands, but that doesn't help us differentiate. So to address this question, Frank Tucci in my lab and Ricky Jodds, a joint student between my and the Hoffman labs, set out to do a very controlled experiment where they would compare the EPR spectra and the cryo-EM structures of parallel samples prepared in these native lipid nanodiscs. And so what they did was they took an EPR spectrum of the PMMO and nanodiscs, and it exhibited 2 EPR signals that we ordinarily would have said, oh, it's copper B and copper C, but maybe it's copper D. Frank then depleted the PMMO of all metal using cyanide, which is a standard procedure in our lab. So that gets rid of the EPR signal. And then he reloaded it with copper 2 into the nanodiscs. And he recovered back this same EPR signal. So our idea was that we could get cryo-EM data on this sample and this sample. We know what the EPR spectrum looks like, and we could actually see, okay, we're adding copper. What site is it going into? What site's occupancy corresponds with this EPR signal? And so that's what's shown on this slide. So the metal depleted PMMO's cryo-EM structure looks like this. And basically this whole region surrounding the copper C and copper D sites is not present in the density. So we can't really say anything about it. However, when Frank loaded the copper back into PMMO and put that into nanodiscs and recovered this EPR signal, order was recovered in the PMOC subunit, and this is what the density looked like. So up top here is just the copper B site, which this is just for a control to show you that this is what it looks like when you strip away most of the metal and then restore it. And then down here is what we see for the copper C and copper D sites upon copper reloading. Again, they're disordered when you take the metal out, so we can't see that. And what we see very clearly is that we have occupancy of copper D, there's the asparagine ligand, and not of copper C. So that tells us that at the very least, in these native lipid nanodisc samples, the EPR signal is attributable to copper going into this copper D site. Okay, so having answered that question and having this more controlled system, we thought maybe we could address the looming question of whether this site, copper D site, is the active site of PMMO. And so we had noticed when we did these first cryo-EM structures that there's this nice hydrophobic cavity right next to the copper D site, and it's lined by these 3 conserved phenylalanine residues. And that suggested to us that maybe this is the place where methane binds. And so now knowing that we have an EPR signal that we can attribute to this copper D site, we set out to see if we could get some sort of a probe in there to tell us if hydrocarbons might bind near that copper D site. And so what Ricky did is he used a product analog, trifluoroethanol, for EPR. So he and Frank made samples of PMMO and nanodiscs with 20-fold TFE. And the reason that we use TFE is twofold. First of all, it gives you the opportunity to observe couplings via fluorine Endor, which is an amazing tool. Second of all, we know it inhibits PMMO. And then I guess third of all, there's no fluorine in the protein. So any couplings we see from fluorine to copper, we know comes from that trifluoroethanol. And so I won't go through all of the data in the interest of time, but this is just a snapshot of the Endor that Ricky acquired. And basically he saw couplings that are attributable to that fluorine interacting with copper 2 and specifically with the copper D. And we know it's the copper D because we've assigned that signal based on these combined structural and EPR experiments. And so over here is, he did a whole bunch of analysis of the hyperfines and the tensors and all that detailed analysis of the Endor data and came up with a model here where the TFE is binding at the copper like this with the fluorines about 5 angstroms away. So that was very exciting. And in parallel, Frank was able to determine the cryo-EM structures of PMMO in the nanodiscs with TFE. And shown here is just the density for the copper sites, the copper B site, and there's another site in this PMMO that I didn't even mention called the BIS-HIS site. And we're comparing them with no TFE and 20x TFE. And you can see that these sites remain the same, and that's consistent for the copper B site with the fact that we don't see any coupling of the TFE to that site. What's shown here is a comparison of the copper C, copper D site region without and with TFE. And with TFE, we very clearly see a new density that can be modeled with TFE binding to copper D with the fluorines about 5 angstroms away. Shown here is that hydrophobic pocket, and you can see that the TFE fits nicely into that region. So this is telling us that this product analog is binding here at the copper D site, and so this is pretty likely to be the active site of PMMO. All right, so I'm going to stop there. Let me just sum up what I've told you. So we were able to get a structure of the active form of PMMO using cryo-EM and native lipid nanodiscs. That showed us a highly conserved and never before observed region of the PMO-C subunit. That region contains a previously unknown metal binding site that we've designated as copper D. And our Endor and cryo-EM data combined indicate that product analogs bind at that copper D site. So with all of those data, our picture of the architecture of this region is complete. We believe this is the active site region, and the kind of next huge question that we would like to answer is, what is really going on with having these two sites here? It's very clear that copper D is occupied in these native lipid nanodiscs, but, we have seen metal bound in this site. And, as a bioinorganic chemist who's been looking at metal sites for years and years, to me, it seems like 2 pairs of histidines should be binding 2 metals, something like that. But we have no evidence for simultaneous occupancy of these sites right now. That and then the nature of the active species are the next frontiers that we're going to address in the lab. So this is the group. I've highlighted Frank and Ricky's work primarily in this talk. I mentioned Chris, who pioneered the Cryo-EM and the nanodiscs. Sue Rowe was the one who started using the membrane mimetics, and Matt Ross, a former joint student with the Hoffman Lab, is the one who really broke open our ability to distinguish these sites by EPR. Also would like to thank my colleague and collaborator, Yuan He, for putting up with all of our Cryo-EM questions over the past few years. And I'd be happy to take any questions.
Thank you, Amy. That is a great talk. I have to give a shout out to Frank Tucci, our CLP training program graduate, and congratulate your entire team on some really great work. It was very interesting to see all the different methods that you use to approach this problem. It's clearly very interdisciplinary in nature. So if you'd like to submit questions either in the chat or, you know, if you want to turn on your camera and raise a hand, that'd be great too. I know it's Wednesday and everybody's tired. Maggie, you're first up.
Hi, I'll ask a question. So thank you for the talk. It was really enlightening and interesting. I was wondering, though, how much structural variability do you think there is in these enzymes across the different kinds of methanotrophs that you introduced in the beginning? You mentioned there were some differences in who had what genes, but in terms of structures or gene components, I'd be curious.
So I think the overall structures, I mean, I know that because we've done structures from other organisms. I think the overall structures are pretty much the same. As I mentioned, that one group doesn't have the ligands for one of the metal centers, so there's going to be some variability there. But again, I think the overall trimeric structure is the same. There may be some variability in terms of their protein interaction partners, which I have not touched on and which we're just beginning to scratch the surface of, but in terms of sort of how they receive their electrons or possibly how they receive the copper, I think based on the genome neighborhoods and what we know about the different types of methanotrophs, there might be variability in that. But I do think the overall structures and the active site location and everything is the same.
Kevin? Looks like you've got your hand raised.
Yes, hello. Thank you for the talk. I appreciate it. I just wanted to ask you about copper B, and I don't know if I missed it or not, but I'm wondering what kind of function you think that may or may not have, it being a soluble in the soluble periplasmic portion, and seeing as the enzymes reacting, you know, oxygen and methane, two gases, if it has any like physical mechanical function, or like if it's involved with cofactors or something like that.
Yeah, it's a great question. So we do think that it's important. You know, anecdotally, you can't mess with it. It's one speculation could be that it's involved in electron transfer, but the problem is it seems to always be copper 2, and we don't have any evidence that it's particularly redox active. Another possibility is that it's structural and it just stabilizes that region, which I don't find that satisfactory, but that's what some people think. So we're still trying to figure that out.
Thank you.
Amy, we've got a question in chat that Lisa's going to read out to you, Lisa.
Hi, Amy. This is from Nicholas Gerst. And his question is, there are methanogen bacteria in the gut. Is there any methanotrophic bacteria in the gut? And feel free, Nicholas, to unmute yourself. And
So I guess it's my understanding that the gut is anaerobic or has not much oxygen. So the methanogens are anaerobic bacteria, and they're making methane. So I don't know that there would be enough air in the gut for the methanotrophs. I mean, there are all these crazy things where people are proposing to, you know, stick methanotrophs on the backs of the cows, so when they exhale, the methanotrophs can eat up all of that methane. Does that answer your question, or?
Sounds like something from a Gary Larson cartoon.
Yeah, there are lots of those.
he says, thank you.
Any other questions? Well, Amy, we'll have to thank you for a great talk and very much appreciate your, oh, Olivia, go for it, please. Yes, thank you.
Sorry, it was just a clap.
Yeah, and I will echo it. Yeah, great talk, and we really appreciate your time today, and all of you for participating. Feel free to suggest to either me or Lisa faculty speakers that you'd like to hear from. We're definitely open to your ideas, but our goal is to share, you know, the wonderful research of all of our CLP faculty out with the broader community. And we'd be very much interested in hearing your opinions on moving back to an in-person format with an actual, we use a whiteboard for the chalk talks, which is always a possibility. The great advantage of doing it online, of course, is that our colleagues on the Chicago campus can participate. But there's much to be said for being in person too.
Well, thanks for having me.
Thank you. And thank you, everyone. See you next time. Bye-bye.
Bye.